Welcome to the Aspiring Geneticist.
Recently, I came across a genetic disorder known as Gaucher disease. It is a rare inherited disorder where fatty substances build up in organs like the spleen, liver, and bones, causing symptoms such as enlarged organs, bone pain, and fatigue. Gaucher disease can affect anyone, occurring in up to 1 in 40,000 live births in the general population. Today, let's focus on type 1. Specifically, what causes this disease? A fatty substance called glucocerebrosidase is a part of a cell's wall and plays an important role in cell activity. Typically, after a cell dies, it is broken down. But in Gaucher disease, they instead build up in white blood cells that are responsible for eating up the debris from dead cells. These atypical white blood cells can pile up in damaged tissues. Well, why isn't it broken down? To answer that, let's take a look inside the cell, the building block of all humans. Now let's imagine the cell containing its own organs like the mitochondria, the powerhouse of the cell, and the nucleus, the library of all the cell's knowledge. The cell is a librarian trying to find the book that contains instructions to build a machine that breaks a fatty substance down. The books are written in the language of DNA, the coding material of almost all organisms. DNA can do this as it is made up of chemicals known as nucleotides. They have four variations in humans at the adenine, thymine, cytosine, and guanine. This is the four-letter alphabet of the cell, and the different arrangements of these nucleotides are what gives rise to blueprints for the tools and weapons of the cell. A nice analogy to how this works is the arrangement of the letters ATR. Try and think up of a word made up of these letters for a moment and comment down below what you thought. I was thinking of a rat, but you could have also been thinking of tar or art. All of these mean completely different things, but they are still made from the same building blocks. Usually, the library has the blueprint for glucocerebrosidase, GCase, an enzyme that basically wields a pickaxe to break down the fatty substance. However, in Gaucher disease, there is an irregular change in the gene, the book for GCase. This mutation is the equivalent of an editor of the book making a mistake in spelling and rearranging words of that book, sometimes even going so far as deleting entire chapters. Understandably, the cell can find the book, but can't fully read it, preventing it from sending the instructions to a factory that can then produce it, leading to deficiency. This is the root cause of Gaucher disease, the lack of GCase from a mutation in the gene that codes for it. The error halts the whole process. However, let us continue to see how the rest unfolds if the mutation was not present. Even if the book was correct, the cell can't just produce it in the library. It must send the instructions to its factory where there are plenty of materials to build it. It can't just take the book out of the shelf though. Imagine the library is more of a messy place where massive structures are built completely out of books. It's like Jenga almost. If you just try to snatch the book out, the whole tower comes crashing down. So, the cell begrudgingly takes out a typewriter and begins copying down every little thing in the book to then mail the pages to the factory. This is the process known as transcription. Specifically, proteins known as RNA polymerase read the nucleotides of the desired DNA sequence and match it with nucleotides of its own. RNA is made up of mostly the same stuff in nucleotides, except that it changes one of the letters, thymine or T, for uracil or U. The RNA polymerase intently reads the book and creates a near perfect copy, in messenger RNA, sometimes abbreviated mRNA. It can actually make a mistake for 1 in every 10,000 nucleotides, but it can correct itself by proofreading its copy. After the copy is written, the cell ships it off to its factory, the rough endoplasmic reticulum, which has tons of industrial machines, the ribosomes, where the message is then used to produce a desired protein in a process known as translation. The ribosome reads the book as long sequences of three-letter words, where it matches each word with a building block, such as a handle in shaped stone. Specifically, the messenger RNA sequence is read in triplets of nucleotides like ACU or CGA, which the ribosome then matches with the corresponding amino acid out of 20 options. Once a ribosome finishes the matching process, the string of letters is now converted into a chain of amino acids that constitute the protein. In this case, translation results in the complete enzyme GCase, fit with its mighty pickaxe, ready to destroy. This is a simplified idea behind what's known as the Central Dogma of Biology, what describes the fundamental flow of genetic information in cells. There are lots of complexities in the system that I didn't cover in this introductory video, but I hope that now you're curious enough to go check it out. Ultimately, even though there may be mutations, biologists have come up with treatments to diseases such as Gaucher's through means like ERT, artificially supplementing the enzyme itself, or SRT, manually decreasing the amount of glucocerebrosidase that the body makes. While not a complete cure, they are both effective in treating symptoms. That's it for today's video. Comment down below what you'd like to see next. Hit like, subscribe, and I'll see you next time.